My name is Carson Roberts from Headwall Photonics, and I'll be talking today about a preliminary study we have done of harmful algal blooms in Santuit Pond on Cape Cod on the east coast of North America. So, talking about harmful algae blooms, I want to first describe what the problem is. And so there are elements necessary to life. If you want to make a potato or a person, the main components you need are carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus. Carbon is important. It's the largest component of most living things. It combines with oxygen and hydrogen to create carbohydrates like sugars and starches and cellulose and hydrocarbons. Hydrogen and oxygen, they're in hydrocarbons and carbohydrates as above. And they're also into hydrogen monoxide, which is in all cells and in all bodies of water, essential to life. Nitrogen is abundant in the atmosphere as the N2 molecule, but that molecule is very tightly bound and isn't biologically available. And it has to be broken down generally into some nitrate form like ammonia. And then it is available for living cells to make amino acids, proteins, and build the rest, many parts of cells. And so nitrogen is very often the limiting factor in the amount of biomass that can grow in any particular environment. That's one of the reasons that nitrates are very important as fertilizers. Sulfur is also essential to life, but it's generally not a limiting reagent in the growth of a particular species. Phosphorus is also essential to all cells. All cells have a cell membrane, which is a phospholipid bilayer. And if you see this picture of all these little cell membranes, every single molecule, every single little ball and stick in that has at least one phosphorus atom in it. And so nitrogen and phosphorus are limiting reagents. An excess of nitrogen and phosphorus in an ecosystem can lead to blooms of otherwise less abundant organisms. So the problem particularly for the harmful algal blooms is human and agricultural waste. Now, human waste, because it takes nitrogen and phosphorus to build people, we excrete them in our waste, and the human waste, if it gets into bodies of water, can increase the amount of biologically available nitrogen and phosphorus and can lead to blooms of otherwise less abundant organisms. Agricultural fertilizers. Again, you're trying to build cells, and so most agricultural fertilizers contain nitrogen, phosphorus, and potassium. So sewage, septic systems, those can put a lot of nitrogen into the atmosphere because although all the solid waste can be collected in a septic system, the nitrates and phosphates are highly soluble and they will seep through the groundwater and get into bodies of water. Runoff from over-fertilized fields can also increase the amount of nitrogen and phosphorus. And so, combined with the fact that we've got global warming, the temperatures of bodies of water are increasing. Most things are going to grow faster when it's warmer. That combined with the runoff from human waste and from agriculture can lead to harmful algal blooms where some species that are toxic can grow out of control and make an entire body of water toxic. And at the bottom, I have two pictures of our study area. Now, these are data that were taken actually with our hyperspectral systems. It's RGB images from the hyperspectral systems. On the left, you can see the pond in November. On the right, you can see the pond in August. It's hard to see in November where our hyperspectral data are because they kind of match the color of the Google Maps. But you can see in August that the pond had turned very, very green. That is the harmful algal bloom, and that is what we are trying to measure to see if we can detect and quantify these with hyperspectral imaging. Now, describe the problem that leads to these toxic algae blooms, which is too much nitrogen and phosphorus coming into an ecosystem. Now I'll talk about the culprit. They're called CyanoBacteria. They're also called blue-green algae. They're not actually bacteria. They're not actually algae. They're very primitive life form. There's a lot of evidence that cyanobacteria are the very first organisms to discover photosynthesis. All plants are green because they have subcellular structures called chloroplasts, which genetic analysis indicates are very likely descended from cyanobacteria. And when cyanobacteria first evolved, they led to a significant change in the atmosphere of the Earth. Prior to the development of photosynthesis, the Earth's atmosphere was reducing. It didn't have much oxygen, had no free oxygen in it. After the development of photosynthesis, the atmosphere changed to an oxidizing atmosphere because it's got oxygen in it. And as all of us know, oxygen is essential to the life of all animals and some plants. And one particular species of cyanobacteria called Chlorococcus is by biomass the most plentiful species on Earth. It lives in the ocean, and there is some evidence that it contributes to about 20% of the oxygen that we have in our atmosphere today. There are many, many different species of blue-green algae or cyanobacteria, and Dolichosporium, Bronychia, and Microcystis all produce toxins that can harm people and animals. And that's what we call the harmful algae in harmful algal blooms. So what is Headwall trying to contribute here? Well, the cyanobacteria, because they're also that are blue-green algae, have different photosynthetic pigments than normal algae. That's why they're called blue-green. And if it's a different pigment, it has a different spectrum. And with hyperspectral imaging, we can look at the spectra of pixels in our images, and potentially detect, oh, this particular spectrum seems to be like the harmful algae, the cyanobacteria, and maybe we can identify them. And the reason this could be useful is these blooms happen slowly. They're not there all the time. I'll show you a little bit about how in the pond that we've been studying, the population of the cyanobacteria rises and falls over the course of the year. And early detection can lead to early mitigation. There are some techniques that people can use to actually reduce the amount of cyanobacteria in a particular ecosystem after a bloom has started. And so what we've decided to do was to work with our partners at Coastal Ocean Vision to get what we call the ground truth. In this case, it's more the pond truth. And they're going to be going through and actually measuring the water at the same time that we are looking from the air with our hyperspectral imagers to potentially come up with a way that we can fly one of our systems over a pond or any body of water and say, yes, there are harmful algae here. No, there are not. Here's where they are. Here's where you should do your mitigation methods. Or this particular beach should be closed because we've detected too much of this dangerous material. So I'll give a little bit of overview of what the Headwall technology is that we're using to do this hyperspectral imaging. And the imagers we use are a Slit-Scan Imager. So on the left, I have a picture actually taken from some airborne data. You can see some striping in it. The aircraft goes back and forth and slowly builds up the image. It builds up the image line by line. So instead of taking the full picture, we'll only take one line of the image, and then we take another frame, which is one more line, and ideally we have some motion that allows us to slowly build up the image. Now, why would we want to take just one line at a time? Next. The idea is that if we take that one line of the image, so that's a row of pixels, something like 640 to 1,000 pixels across our detector, we disperse that so that each one of those image pixels is re-imaged at a different wavelength, so that each pixel then has a spectrum. And so we take this single slit and we use the entire detector to get this full spectrum from every slit. And then we build up our image that way. And then we build up this hyperspectral image where each pixel has the spectrum. So how does the imager work? What we do is we have a 4-optic that makes an image onto a slit. Again, that's this first focal plane that's mostly dark, but transmits light just over a single row that's about the same size as one of our pixels. It is re-imaged onto an electronic focal plane array with a 3-mirror relay. One of those mirrors is a diffraction grating. And so because the diffraction grating reflects different wavelengths of light at different angles, we end up with the image I showed you in the last screen, which is one line of pixels re-imaged at many different wavelengths, typically on the order of 300 different wavelengths. And so each time we take a frame, we get, in the case of the imager that I'll be talking about today, 640 spatial pixels and 270 spectral pixels. And then we take another line, we move forward, take another line, we move forward, and we build our image up that way. So in addition to the hyperspectral imager, the research we did also, our payload included a LiDAR, just like radar, and it's light detecting and ranging. The instrument has 16 paired photodetectors and lasers. Each laser fires, and then the time to a return detected by the photodetector is measured. Because light travels at a constant speed, if you measure the time it takes for the light to get there and back, you know twice the distance to it. And then the lasers are spinning around. We're taking thousands and thousands and thousands of these per second. And then that gives us these points in space. And each point has a latitude, a longitude, and altitude, and some other metadata, including, and this is important for this research, the GPS time at which that point was collected. So on the left, you can see a picture of the drone that we used for some of this research. It's a DJI M300 with the Headwall Nano Hyperspec and a LiDAR on it. And it flew back and forth over the pond. And then in the right picture, you can see a small boat that has the detector that is actually sampling the water and measuring and counting the number of various species of plankton in it. And you should be able to see that pond is green. This was in August, and this was when the pond really was toxic. Fishing was banned, swimming was banned, and there was a tremendous amount of the cyanobacteria in the pond. So next, I want to talk a little bit about what our colleagues are doing in the boat. So they have a Raman imaging flow cytometer. And so the flow cytometer, cyto, I believe, means a cell, so it's a measuring cells. It's sucking up water. It's running it through a microfluidic channel, and then it gets into a little measurement chamber. And if there's a cell detected, a laser blows onto it, and it gets a Raman spectrum from that. And the Raman spectrum, similar to the reflectance spectra that we're getting from sunlight, could give you a very accurate measurement of what the pigments are, and that can be used to distinguish the various different species. They're using some sophisticated computers to figure all this out. And the net result is the folks from Coastal Ocean Vision are going back and forth over the pond in a boat. They are measuring the relative concentrations of all the different plankton species, in particular the harmful algae, at the same time that we are flying over them. Next, please. And so this picture, if you see in the left-hand slide, there's something that looks like a aluminum tube, because it's an aluminum tube. And inside of it is another version of this Raman imaging flow cytometer. And in the boat there, you can see our colleagues. They went back and forth over the pond. On the first image on the left, you can see before the flow cytometer is fully deployed. On the right-hand side, you can see it drags along in the water, and it's constantly sampling the water and geotagging and time tagging all the measurements that they're getting of the relative abundance of the various different plankton species. So here's another picture. We were out there in August. The toxic algae blooms tend to be correlated with large rain events because that increases the runoff, and that in turn increases the biologically available nitrogen and phosphorus that can lead to blooms of these toxic algae, including Dolichospermum. And you can see this a picture. They seem to form little rings. The cells are quite small, but there's enough of them that they really, really color the water. So in addition to this HAB stats system that they're dragging around in a boat, there are 4 or 5 buoys that sit in the pond, and they measure the relative abundance, in particular of the Dolichospermum harmful algae, as a function of time. And I've got 3 red arrows there. We did our first flight over the pond, along with our colleagues in the boat, in June. We did another mission in mid-August, and we did one more mission in November. And you can see from the blue lines in the upper left-hand side that there were very few of the toxic algae in June. There were few also in November, and our mission in in August corresponded with near the peak of these toxic algae. And then one of the things that seems to affect the population of these harmful algae is chytrid fungi, which my understanding is they latch onto those little rings of the Dolichospermum and cause them to sink to the bottom and die. And there was a bloom of these chytrids, which led to a crash in the population of the toxic algae soon after our mission in August and well before our mission in November. So I'm jumping a little bit ahead here, but this is a map where I have mapped 3 different types of toxic algae based on their reflectance spectra, and I've circled several locations of the boat. So we're flying over the boat, and we can actually find the boat in the imagery. Because we have spectral imagery, we can take the image and we can classify each pixel according to what its spectrum is most like. And so I've built a library with several different kinds of trees, a couple of species of Elodea, which is a non-algae plant that grows in the pond, 3 different types of toxic algae, and 3 different spectra that I was able to associate with the boat in the imagery. So the next slide shows some of these images. So RGB images taken from our ortho-rectified mosaics of the hyperspectral images. And then below that, classified images. It's all pink because they are going through a soup of the toxic algae. And then that's how I can identify, oh yes, there's the boat there because I can see the fiberglass and the canopy and the white froth from its wake. And since I have the LiDAR, the LiDAR measures altitude. Well, the boat is on top of the water. The water is flat. And so anything that sticks up above the water can be seen in the LiDAR point cloud. All of these little things that look like little worms in our images. On top of, you can see sort of a grid pattern where we went back and forth, north and south and north and south and north and south. And you can see there the path of the aircraft. And it took us a little while to coordinate well with the boats, but by the end of our mission, we got 4 different images where the boat was very clear, and I can get the exact GPS coordinates and the GPS times for each of those boat locations, and we can use those to very carefully correlate the data that were taken by the boat at those exact times. Next. And so here then are the times and the GPS locations for them. This particular image, instead of being colored by altitude, is colored by GPS time, with blue being the beginning and red being the end. And so that's how I can extract from the LiDAR the actual exact locations and times of the boats for correlating these measurements. Okay, so here is a part of the study area where I've overlaid it on the Google Maps. The Google Maps, I don't know when these images were taken, but not during a toxic algae bloom because the pond doesn't show a lot of reflection. Water doesn't reflect a lot of light unless it's got a lot of stuff in it. And it had a lot of stuff in it in August. And you should also see that there's some lighter and darker areas here. That's because we're doing this, we're doing all these measurements where all our light is coming from the sun. And so if a cloud goes over, it gets a little darker. And if we get bright sun, we get a little brighter. And these intensity variations can have some effect on the classifications, but not a lot because the method that we're using to do spectral classification, which we call spectral angle mapping, is somewhat insensitive to the absolute intensity of the spectrum. Okay, so this is not images from the pond. This is some other work that we've done looking at in shortwave infrared spectra of geology. And this is an example of one way that we can do the spectral classification is to build up a reference library based on the ground truth. So in this case, this is out in Culprit, Nevada, and we flew over this area, and our colleagues, the geologists, went out and collected a bunch of samples. And they brought those samples back to the lab. They used their geological techniques to identify what the mineral composition of each of their samples were. And then they took spectra of them from a lab spectrometer. They gave me the spectra, and then I used those as a reference library to compare to the spectrum in each of the pixels and say, okay, this spectrum, this pixel right here, I'm going to color it yellow because it's gypsum. I'm going to color it red because it's montmorillonite. And so that's one way of doing the classification. And when we continue this research, we will probably be doing this with some spectra taken from samples taken from the pond. But for this preliminary work, we did another method, which I'll try and outline in the next slide. And that is, I identify from the RGB image, I can say, this is very clearly a tree. This is very clearly the algae in the pond. And if you look towards the middle of the image, very near the shore, there's some little stripy areas where it seems much brighter than others. And so I chose 3 different classes of the harmful algae, 3 different classes of trees, and built up a spectral library, not from ground truth, but from pixels in the image. And so I chose a region that looked relatively uniform, got an average spectra from a few thousand pixels of that, built up the library, and then used that library to then classify all the pixels in the image. Okay, the next slide, please. And so here are a couple of images of the reference spectra. Anybody who's familiar with the spectral biology will recognize in the spectra of the trees what's called the red edge. And that's because chlorophyll, which is one of the main pigments in the blue-green algae and in trees, has a very strong absorption in the visible, but there's not enough energy in infrared light to drive photosynthesis. And so the chlorophyll pigment doesn't reflect past about 700 nanometers. And then you get the reflection of cellulose. And if you think of a piece of white paper, that's almost 100% cellulose. It's very reflective of light. And so you can see in the various trees, a lot of absorption, except in the green, around 550 nanometers. You could see a bump there. And then a high reflection out in the infrared. And then when we zoom in on these lower levels, you can see that there is significant difference. In fact, a strong bump I see around 650 nanometers that's much more associated with these toxic algae than it is with the trees or with the Elodea, which are the flowering plants that live in the pond as well. Next, please. So let's go. Again, we flew this pond 3 times, once in June before there was a lot of toxic algae. But if you look at our imagery from June, there's a few bits of pink there. So there was some, it just wasn't a huge bloom. And part of the reason that there wasn't a huge bloom, there was a lot of this Elodea. Now, Elodea is a plant that I was first familiar with from aquaria. It looks sort of like a feather brush, long strands. And in the pond, it made these large mats of it. In fact, when our colleagues were driving around in the boat, they kept getting stuck because there was so much of this Elodea. And one theory is the toxic algae bloom was late this June, and that may have been because there was so much of the Elodea that it sucked up all of the available nitrogen and phosphorus, or a significant fraction of the available nitrogen and phosphorus, and delayed the toxic algae bloom. But based on our spectral classification, remember, I've made these toxic algae various colors of pink. There is some, particularly around the shoreline, and it seems also associated with the edges of these Elodea. So let's go to the next one. Now we have superimposed on our spectral classification data from the boat driving around. And we've got the Dolichospermum, the Microcystis, and then non-toxic green algae. And they're not getting a lot. These dots are associated with the concentration in number per milliliter. but there's some correlation of areas where we are seeing a little bit of pink in our images, and they're seeing some of these, some high concentrations of these toxic algae, but not yet a bloom. Okay. Now we go to August, where there was a lot of the toxic algae. And if you look at the RGB image, towards the center, I see a bit of a line heading off to the northeast. And those seemed different enough to me that I decided to make them as different spectral classes. And then we do the classification. It really looks like there is a differentiation. There's two clear spectral classes of the HAB in the pond. And if we compare that now with the data taken by the sampler in the boat, we see very clearly, again, on the eastern side of the pond, it looks as though the toxic algae is, the population is dominated by the Dolichospermum species. And on the on the west side of the pond, it's dominated by the Microcystis species. And this is, to me, the most intriguing result of the whole thing, is that it appears that we can distinguish between toxic algae species just by the spectral characteristics as measured by the drone flying over the pond. And so this is preliminary work, but this indicates that this is something that we will pursue next year with more studies and more ground truth. But again, it really does look like we've proven out the idea that we can see the toxic algae, and we can identify different species just by their spectral reflectance. And to complete the study, let's look at the data from November. So after November, remember I said there was those fungi that seemed to have caused a crash in the population. We fly over, again, the pond's not green, but our data seem to indicate that there are still some toxic algae, and most intriguing to me is in the northeast corner. In the top of this, we see a small area that's quite bright pink. That happens to be where a stream that is flowing through a cranberry bog, which is fertilized, is emptying into the pond and leading to more of, a small bloom of this toxic algae. And if we again look at the correspondence, again, up there where I see that little pink thing, big red ball, but the rest of the pond is mostly populated by the green algae. And so that's our basic results. Very encouraging. We will be working next year with our colleagues at Coastal Ocean Vision to try and extend this research. And I would like to acknowledge our main pilot is my colleague Charles Kepler. Scott Gallagher from Coastal Ocean Vision ran the boat, did all the work. My colleague from our marketing team, Ross Nakatsuji, organized everything for us. And the rest of the people at Headwall, the people who build the imagers, the people who do all our work. And there were several of our colleagues who were with us. These missions generally take a bunch of people to get the drone in the air and get everything going. And thank you very much for listening to this talk.